Dear children, in this module, we are going to learn about the endomembrane system, which is comprising of the endoplasmic reticulum, the lysosomes, the Golgi complex, and vacuoles. What is this endomembrane system? It is a group of membrane-bound organelles, membranous organelles having coordinated functions, that is the endoplasmic reticulum, the Golgi complex, the lysosomes, and the vacuoles. They function together. They are different cell organelles, but they function in a coordinated manner. So that is why these four together, they form the endomembrane system. Now let us learn the first part of the endomembrane system, that is the endoplasmic reticulum, which is represented as ER. These endoplasmic reticulum are a network. It is made up of a network of tiny tubular structures which is scattered in the cytoplasm. It is made up of a network of tiny tubular structures scattered in the cytoplasm. The endoplasmic reticulum, which is made up of a network of tiny tubular structures which is scattered throughout the cytoplasm, it compartments the intracellular space. Intracellular space means the space within the cytoplasm or within the cell. Intracellular space is the space which is seen within the cell. It differentiates the intracellular space into two. That is the luminal space and the extraluminal space. What is this luminal space? It is the region which is seen inside the endoplasmic reticulum, and the extraluminal region is the region which is seen outside the endoplasmic reticulum, that is the part of the cytoplasm. Now there are two types of endoplasmic reticulum. They are the rough endoplasmic reticulum and the smooth endoplasmic reticulum. The rough endoplasmic reticulum is represented as RER, which stands for rough endoplasmic reticulum. Why are they called as rough? Because they bear ribosomes on their surface. We can see ribosomes on the surface of this endoplasmic reticulum. So those endoplasmic reticulum which has ribosomes on its surface are called as rough endoplasmic reticulum, and is frequently observed in cells which is actively involved in protein synthesis and secretion, because we know the function of ribosomes is for protein synthesis, or it is helping in translation. So those cells which is actively involved in protein synthesis or translation are having this rough endoplasmic reticulum. They are extensive and continuous with the outer membrane of the nucleus. We know this is the membranous part of the nucleus. The nuclear membrane is divided to outer and inner. The outer membrane is facing towards the cytoplasm. So this rough endoplasmic reticulum, they are extensive and they are continuous with the outer membrane of the nucleus. Whereas SER, which is called as the smooth endoplasmic reticulum. In these smooth endoplasmic reticulum, they are smooth in appearance. Why? Because they are lacking ribosomes and is a major site for synthesis of lipids, whereas rough endoplasmic reticulum is helping in the synthesis of proteins, whereas smooth endoplasmic reticulum is helping in the synthesis of lipids. In the case of animal cells, it is having an additional function. The SER is having an additional function. It is helping in the synthesis of steroid hormones. Okay, so the smooth endoplasmic reticulum is involved in the synthesis of lipids and steroid hormones, whereas rough endoplasmic reticulum, which is having ribosomes on its surface, is helping in the synthesis of proteins. So that's the function of endoplasmic reticulum. It is helping in the synthesis of lipids and proteins. Now let us learn about the second organelle which is forming a part of the endomembrane system, that is a Golgi complex, or it is called as the Golgi apparatus. The Golgi apparatus or Golgi complex is densely stained structures, reticulate structures, which is found near to the nucleus. Not only near to the nucleus, it is found in close association with the endoplasmic reticulum. The Golgi complex was first observed by Camillo Golgi in the year 1898 and hence the name Golgi complex. And the Golgi complex is made up of flat, disc-shaped sacs. These disc-shaped sacs are called as cisternae. They are made up of flat, disc-shaped sacs called as cisternae. And these cisternae are 0.5 to 1 μm in diameter. And these cisternae, they are stacked one above the other. And these cisternae, they form concentric, they are arranged concentrically. These cisternae, they are arranged concentrically, and this arrangement is having a peculiarity. It is having two phases. It's having a cis phase and a trans phase. The cis phase is called as a forming phase, whereas trans phase is called as the maturing phase. Okay, the cis phase and the trans phase. Cis phase is convex. The cis phase is convex, whereas the trans phase is concave. So the cisternae, which are stacked one above the other, they are arranged concentrically to form a cis phase, which is convex, and it is the forming phase, and the trans phase, which is concave and which is a maturing phase. Both the cis and trans, they are different from each other, but they remain interconnected within this Golgi complex. Now, what is the function of the Golgi apparatus? It secretes certain materials. The first function is secretion. So it secretes certain materials which are received by certain intracellular targets, that is within the cells. And also these secretions are needed for outside. It is also transported outside the cell. And second is function is packaging. So materials to be packaged as vesicles, which is released by the endoplasmic reticulum. They fuse with the cis phase. So materials which are released from the endoplasmic reticulum, that is steroid hormones, then the lipids and the proteins, which are synthesized by the endoplasmic reticulum and which has to be packaged in the form of vesicles. They move or they fuse with the cis phase, which is the forming phase. And then they move towards the trans phase. From the cis phase towards the trans phase. From the cis phase, they move towards the trans phase. And that is why just now I told that it is seen in close association with the endoplasmic reticulum. So substances which are released by the endoplasmic reticulum is packaged in the form of vesicles by the endoplasmic reticulum by passing through the cis phase and released out to the trans phase. And again, the protein which is synthesized by the ribosomes of the endoplasmic reticulum, they have to undergo modification in order to be functional. And this modification is done in the cisternae of the Golgi apparatus before they are released from the trans phase. So within the Golgi apparatus, the proteins which are released by the endoplasmic reticulum, they undergo modification. And it also helps in the formation of glycoproteins and glycolipids. So these are the functions of the Golgi apparatus, that is secretion, packaging, modification of proteins and formation of glycolipids and glycoproteins. So thus in this module we learned about two organelles of the endomembrane system, that is the endoplasmic reticulum and Golgi apparatus. Thank you.